All right, thank you so much for being here, guys, and good morning to everybody. I really appreciate this nice introduction and, of course, this opportunity to present my research in such an organization where I'm originated from, basically. So it's a really proud moment for me. Anyway, so without further delay, I'm going to talk about, as it is mentioned, one of my PhD research project that is on finger millet salinity tolerance using the metabolomics. So before I go to my next session, I would like to say you something regarding the conferences. Attending conference in the United States as a graduate student is really a great opportunity. And for me, it was Crop Science Society of America meeting back in 2022. It was in Baltimore, Maryland, and for the first time, I saw such a huge event. There were Agronomy Society of America, Soil Science Society of America, as well as Crop Science Society of America were involved, and it was a great experience. I was presenting a poster there. It was also, a preliminary study on finger millet back then, and I was presenting that research. Interestingly, many students and researchers were coming to my posters. I thought that I have done a remarkable, research. That's why people are very interested to know about it. But do you know, in that evening, which questions I answered the most? Many people were just asking, "What is finger millet?" Because this crop is so new to the United States and the acreage is so limited that many people even didn't know what is finger millet. I know this situation is little different in India. We know what is finger millet. We have seen finger millet, but in the United States, it was a different story. So it was very funny for me that I just answered what is finger millet, not my actual research. So today, if there are anyone like that who don't know about finger millet, for them a little brush up on finger millet, and then I'll talk about problem of salt, why you should care about, then my research objectives, materials and methods that I have used during my study, and finally I'll wrap up this session using my talking about my results and discussions. I hope sounds good. All right, let's talk about finger millet. So finger millet, which is Eleusine Coracana subspecies Coracana, is an allotetraploid crop having two genomes, A and B, with 36 chromosomes and 1600 megabase pair of genome size. It is predominantly self-pollinated crop, so it means a little lesser, complexity in genomes than other crops, but less recombinations as well. So we don't have much variations. It grows well in a crop in a situation like 30 degrees Celsius that is warm, and of course the rainfall of around 1,000 millimeter. It was predominantly grown in the countries like India, Africa, but it was basically originated from Ethiopia and Uganda region. And then it was brought into India. Right now, it is also spread in Bangladesh and Nepal region as well. However, if we talk about now the finger millets in a nutritional quality, if you're buying 100 gram of finger millet, you're getting 73 gram of carbohydrate, which you might get from rice and wheat or like other traditional crops, maize as well. But you are getting an extra 11.5 gram of fiber and 5.5 gram of calcium, magnesium, vitamins, which are very, very essential and very important for our health, right? So this is why the finger millet is a good fit for such a country like India, where population is a big challenge, right? And agricultural scientists always face the problem to feed their ever increasing population with nutritious food. For such kind of situations, finger millet can be a very good fit, but we need to increase the agricultural extension part. We have to let the people know, we have to let the farmer know its potential, its nutritional quality, in a good situation, in a proper way. So anyway, so for this kind of countries, if we go for a big cultivation, abiotic stress tolerance we need, because this is a global problem. And talking about other like drought, heat, salinity is also a global problem. But whenever we think about salinity, one thing we always think that is the coastal areas are probably vulnerable most, because we think that the water intrusion from the sea makes the soil salinization. But for those who are thinking in that way, a big warning for you, because there are several other ways, other factors that can also cause soil salinization. For example, prolonged use of recycled water in the irrigation. For example, the water which is coming from the industrial waste or from the household, that may contain a lot of salt in it, right? And if you keep irrigating your land for the prolonged time with this kind of water, that salt will be deposited on the soil and can cause soil salinization. And this situation is even worse when it comes with the global warming. When the weather is very warm, in that situation, water gets evaporated, leaving the salt in the soil intact. So over time, it gets deposited, and it can cause soil salinization as well. And another factor is very common, that is the injudicious application of fertilizer. Now, if you think, what are the fertilizers? Fertilizers are nothing but the complex form of salt. So if you overdose it, you are overdosing salt. That is going to be deposited on the soil and can cause soil salinization. But to understand now the physiological effect on the plants, let's talk about one thing. If I now ask you one question, that do you need salt in your food? You might say, of course I need, but up to a certain limit, right? If it goes like beyond that limit, or like if I make too much salt in our food, then you might say that no, I don't want any kind of the very salty food. So, but as a human being, you have the privilege to say no to salt. But think about plants. Once they start accumulating salt in its root zone, they are very helpless unless we help them. Eventually, they can because they can move, and eventually they die due to the salt problem and the physiological consequences. Now we will discuss what are the physiological consequences under salinization. Anyway, now if we talk about physiological consequences, first thing comes the water uptake in plants. Under normal situation, plant is having lower water potential than soil. This psi describes or denotes the water potential, right? So water potential of soil under normal condition is higher than plant. As a rule, it facilitates water uptake from the soil to plant because water will flow from higher to lower water potential. But under salt condition, when the plant is having a lot of salt in its rhizospheric region, now the situation is reversed because the water potential of the soil is now depleted than the plant. As a result, plant is having now the higher water potential, and as a consequence, water will flow from plant to soil. So that causes the osmotic stress to the plants. But to keep continuing their life, the plant has to uptake the water somehow using osmolytes or whatever, lowering their water potential. They have to uptake the water. But this water is now contaminated with salt. So once the water uptake continues, the cells actually accumulates a lot of ions. For example, if you talk about sodium chloride, sodium ions gets included or incorporated into the cell. That causes the cationic competition with other essential compounds like potassium, calcium, and magnesium, which causes the efflux of such essential components and can cause ionic stress. This situation is even worse when it goes further and causes the reactive oxygen species. These reactive oxygen species can break down, like macromolecules like carbohydrate, proteins, and fats. And if you think about the cell membrane, which are composed of proteins, lipids, and carbohydrates, and many other cellular components, I know, those are made of those macromolecules. Once those are depleted or broken down, that cell electrolytes can passes through those leakages, and eventually the cell can die, and eventually the plant will die, right? Now, for the objective of my research, I had two lines, IE405 and IE518. I had to check them morphophysiologically under salt condition and as well as the metabolomics. Now, for morphophysiology, it's because we wanted to see what are the physiological response there under the salinity tolerant salinity condition, sorry. And the metabolomics part to understand their mechanism, the salt tolerance mechanism. There are several reports available on salinity, variation among finger millet lines, but none of those really talk about the metabolomics part or the metabolism which are getting altered. Anyway, here the two lines which you see, that IE405 and 518, were not the random choice. We had a previous experiment with 12 finger millet lines at the germination as well as the vegetative stages. And from that, we found that these two lines are consistently having contrasting, performance under salinity. One being tolerant, another being susceptible. Now you might be wondering which one is what, right? We will see in the next result section. For now, the materials and methods section, we germinated our seeds in the greenhouses, and then after 14 days, it was transferred to the growth chamber where we control the temperature, humidity, as well as light conditions according to the requirements of the finger millet growth conditions. It was a split plot in RCBD, the experimental design, and there were three replications with two salt treatments, one being control, another being sodium chloride. And in each whole plot, we used the genotypes randomized. For IE45 and 518, each of those were having 4 plants in it, in each whole plot. Now, talking about the screening of the plants, we used several different, morphophysiological parameters like plant height, photosynthesis, and FVFM is the maximum quantum yield of the photosystem 2. It is a bypass, measurement of the photosynthesis. higher values of photosynthesis, higher value of FVFM, higher value of plant height, as well as the dry biomass indicates better tolerance. However, now we also considered the sodium and potassium ion content there. Higher sodium ion content is detrimental for plants, whereas higher potassium ion content is good for plants. So under salt condition, what generally happens, sodium content gets higher because we are supplying the plants with additional sodium content, and potassium content gets depleted. Electrolyte leakage and MDA content, both are product of the oxidative stress, which we talked about, that reactive oxygen species breaks down the cell membranes or the carbohydrates, proteins, fats, and that is why we got the electrolyte leakage as well as malondialdehyde, which stands the acronyms for the MDA. And MDA is the byproduct of the lipid peroxidation, and once the lipids have got broken down, MDA content starts, the plants actually starts accumulating MDA. So which means higher electrolyte leakage, higher sodium ion content, and higher MDA content, all are detrimental to plants. For today, we will only consider these three measurements: sodium ion content, electrolyte leakage, and MDA content. It's because they have more pronounced effects on metabolism. For the statistical analysis, we used a mixed model where the genotype treatment and genotype cross treatment were used as the fixed effect, and the random effects was the whole plots. And further, the means were separated using Fisher's protected LSD at a P level of 0.05. For the metabolomics, we collected the leaf samples, freshly harvested, and then it was freeze-dried or the lyophilized, and after that, it was sent to Georgia Tech University for isolation of the metabolites using ultra-high performance liquid chromatography and mass spectrometry for the detection of the metabolites. For the metabolic analysis, we used three different analyses. First, principal component analysis, fold change analysis, and pathway analysis. We'll discuss all of those. Now talking about the results and discussions. First, the morphophysiological results. Electrolyte leakage and MDA content, both are detrimental to plants, but under control conditions, since the plants were not experiencing any sort of stresses, both plants did not show any significant difference. It means they are performing the same way. But under salt condition, it is clear to see that there was significant difference, as well as the IE405 is having much higher amount of MDA as well as electrolyte leakage, which indicates that IE405 is the susceptible line towards salinity, and IE518 is the tolerant line. Now talking about the sodium ion content. As we expect under control condition, there were no significant difference, but interesting story is now. Under sodium ion concentration was higher in the susceptible line, which you expect, that IE405, but there were non-significant effect found in the salt condition for both genotypes when you consider the potassium ion content. This situation is even more interesting if we just pay attention to the salt condition. Think about the plant's growing situation. Plants were grown with added sodium chloride. There were no additional potassium. Whatever the potassium was there, it just in the fertilizer, not an extra amount. But still, the above ground part is having much higher amount of potassium than the sodium. So the question is, where did that sodium go? And this is above ground part only. We didn't check the sodium ion content at the lower ground part of the root. So two hypotheses comes in mind now. One, either the salt is compartmentalized somewhere in the roots, or it is sequestered in the roots, or it is excluded from the root zone to the rhizosphere again, so that the above ground part did not get a chance to accumulate excess amount of sodium. That's why it's still not clear to us, and we are trying to find out the actual answer to it. But these are two hypotheses for now. And this is right now we will move on to the metabolomics part. And when I first got my metabolomics data, I actually had 15,427 rows in it. So Excel sheet that is more than 15,000 rows. You can understand how huge that the data set was. So first we wanted to see that whether they have a differential metabolite accumulation or not. And we, under principal component analysis, we saw under control condition they have no significant difference, whereas under salt condition a clear differential pattern we found. Now the thing was to check the fold change analysis. That is the indication of the how many metabolites were differentially accumulated in one line than others. Now onwards, whatever I'll be talking about, it will be based on the comparison of the tolerant line in comparison to the susceptible line. Here, our tolerant line is 518. So I'm comparing 518 versus IE 405. Under control condition, we saw that the tolerant line upregulated 124 metabolites and 206 metabolites were downregulated. But under salt condition, the numbers were much higher. 581 metabolites got upregulated, whereas 468 metabolites got downregulated. So you can understand that how much salinity was playing effect on its metabolism. This is the significance. That from 124 metabolites upregulation, it got higher to 581 and downregulation from 206 to 468. Now, next step was to find out how many of those 581 metabolites under salt condition were also present in the case of the control conditions. Similarly, how many of those 468 metabolites were also present in the control conditions? Why it is so? Because think we are trying to find out the metabolism, alteration of metabolism under the salt condition, not control. So we wanted to discard those metabolites which are present in both condition, control and salt. We don't want them. We just want the salinity-induced metabolites, either it is upregulated or downregulated. Okay, now if you see this Venn diagram, it is clear that 32 metabolites in that comparison, 32 metabolites got commonly upregulated under both treatment conditions, and it was 67 metabolites under downregulation under both conditions, salt and control. So we discarded those, and we moved forward with 549 metabolites which are upregulated and 401 metabolites which were downregulated, uniquely due to salinity, not something else. After this, we uploaded these metabolites into a software called MetaboAnalyst, and then we tried to identify the key pathways which are getting reflected or influenced in the tolerant line. And we saw that there are several different actually pathways got upregulated and downregulated. But here, what we will be focusing on, that butanoate metabolism, which is responsible for the stress signaling. To start any of the plants, to start any kind of gene regulation or anything after any stress, plants needs to know first. They need some information first. That is called the cell signaling, and butanoate metabolism is responsible for cell signaling, which got upregulated in the tolerant line. Now to continue the processes, the metabolic processes, a lot of energies are required, right? So energy will come from the respiration or photosynthesis. Now, if you see that citric acid cycle or the TCA cycle and pyruvate metabolism both got upregulated in the tolerant line. And other than that, as I mentioned, that due to the oxidative stress, macromolecules got broken down. That includes proteins and fats. To reconstruct those proteins, we need amino acids. So that we found several different amino acids got upregulated. That pathways got upregulated in 518. Now, one thing is I want to make clear here. Here, whatever what I'm showing here in this slide, those are pathways. They are not the individual metabolites. It means the network of metabolites, which is reflected in a particular pathway. So for example, TCA cycle is having a lot of different metabolites. Those are significantly upregulated and downregulated, and based on that software gave us the idea that, okay, the TCA cycle is upregulated intolerantly. But in the next slide, which I'm going to talk about, it will be on individual metabolites, not the pathways. So here if you see, under both control and salt condition, but we will mostly focus on the salt condition, since the control condition, we didn't see much significant difference among these two genotypes. So the green line or the green shade is showing the downregulation of metabolites, and the purple one is showing the upregulation of metabolites. So in this case, again, the 518 is having, higher upregulation of some sugars. Here are several different, but I don't have time to go over all of them, but I will talk about some simple ones, which will be easy to understand, like mannose, millibase, L-proline, and gamma-aminobutyric acid. All of those acts as osmolytes. So what are the functions of osmolytes? Osmolytes are nothing, just once they accumulated in the tissues, they lowers the water potential of the plant. Now, if you remember, I talked about the osmotic stress in the plants, right? Under salt condition, soil water potentials got depleted due to the salt contamination. And in that case, plant was having higher water potential. So now, if we want to facilitate or if we want to reverse this situation, reverse this equation, the plant has to have a much lower water potential than soil. That means more negative water potential than soil. That comes through the accumulation of osmolytes. Once these osmolytes, that is the L-proline, GABA, millibias, mannose, this kind of osmolytes gets accumulated in the plants, this situation is reversed now. The water potential of soil again higher than the plant. Now, as a rule, water will flow from higher to lower water potential, right? So water can now again, pass through the soil and can go to the plant, which means the plant is making the osmotic adjustments using those osmolytes. So, which is evident from the tolerant line as well. That is maintaining better osmotic adjustments, better energy metabolism, better signaling molecules, and as well as lot of amino acids, which will reconstruct their proteins. So those are the key metabolic pathways that got upregulated, or the metabolism that got upregulated in the tolerant lines. Now, in summary, we saw that the lower electrolyte leakage, lipid peroxidation, and sodium ion content in shoot tissues in the tolerant lines, which is the morphological evidence that IE518 is performing better than 405 under salinity condition. Next one, metabolic analysis, as I mentioned right now, that osmotic adjustment, stress signaling, energy metabolism, and amino acid biosynthesis got upregulated in the tolerant line. And finally, sodium ion concentration, which is I don't have the answer yet, but the very interesting finding here that the sodium concentration of the above ground tissues were much lower than the potassium ion content, but even when the plants were supplied with the additional sodium chloride, but no potassium. So the next question is, where did that sodium go? Either it was compartmentalized in the root, or it got effluxed from the sequestered from the root zone, actually to the rhizospheric region, so that the above ground part were saved. So probably in some few months, I will be able to find out that result, that dilemma. And with that, thank you so much. I would like to acknowledge my major advisor, Dr. David Jesperson, Dr. Paul Remer, my committee member, Dr. Katrin Davis, and Dr. Brian Schwartz, and of course my lab members, NSF for providing the funding, that is the National Science Foundation of America, University of Georgia, USDA for providing me the seeds, and Georgia Tech for the metabolism part. And of course, Visva-Bharati, পল্লী শিক্ষা ভবন, এটা আমাকে সবসময় একটা খুবই আনন্দের ব্যাপার যে আমি এখানে একটি করছি বিশ্বভারতীর জন্যই, and because of Visva-Bharati's invitation, I could make this. And I really appreciate all of your kind presence and patience here. If you have any further queries, you can reach out to me using my email ID, and you can also scan this QR code, which will take you to my LinkedIn profile. With that, thank you so much for being here, and I really appreciate all this opportunity. Thank you.